Very good evening, everyone. Today, dual purpose is going to be served in our lecture. I'm going to talk about the practical aspect of biology because every subject is having practical aspect, and if you don't know about that, it will be difficult for you to deal with that. Okay. Now, what practical we are going to study? In your practical examination, you will be having questions about spotting. Even if it is online practical, offline practical, Gujarat board, CBSE board, in all boards, students will have this type of experiment of spotting. So what is expected in case of spotting? In case of spotting, they will display you either specimen or the picture or the slides in which you can see the organism under microscope to you. And then what you have to do is you have to simply identify the organism. You need to mention in which category they belong to. You need to write one or two unique characteristics of that organism, and then you can draw the diagram in exam. Okay, so spotting is carrying very high marks in practical examination and it is very scoring. You may think कि मैम इसमें क्या बड़ी बात है मैम, ये तो हम कर लेंगे. But when you look for an organism under microscope, they look quite different. They are not diagrammatic always. So today I will give you an introduction about the microscopy. Okay, and then we will move ahead with the spotting of plant and animal kingdom. So are we ready to start?
Yes, ma'am.
Yes. So we will move ahead. So today the first topic of discussion will be microscope. It is one of the most important instruments which should be part of life of every biologist. So I am going to display the diagram and talk to you regarding it. Okay. So here are different parts. You need to tell me what are the parts and what are their purpose. So we will start with this one. What is it? Class, this J. Anyone wish to try?
Ma'am, mirror.
Yes. What is the role of mirror?
Ma'am, it reflects light, which helps to make the organism more clearly visible.
Reflects light for seeing specimen. Okay. Then is this E part. This E part, what is the role?
And base.
It is base. What is the role of base?
To give support to microscope.
Or it is holding other parts? Okay. So what is this I?
Ma'am, condenser.
No. K is condenser. What is I?
Ma'am, cork-like structure which hold the condenser.
No. It is called iris or diaphragm. The role of iris or diaphragm is it controls the amount of light reach to light. What happens is it is a membranous structure. If you look into condenser lens, this structure is a membranous structure. If it is partially closed, then this much light can enter. If it is half closed, then this much light. If it is completely closed, no light can enter. And if it is completely open, highest amount of light can enter. So this is a thin membrane which you can control. There is a knob. I will show you a very wonderful video after this, which will show you 3D structure of microscope. By twisting it, you will be having good idea about it. Okay. So this one is condenser. Okay, the K is condenser. So what is this condenser do? What's its role?
Ma'am, help to condense the light rays.
So help to condense means what? The meaning of condensation of light I wish to share with you is, consider whenever light rays are coming from a source, they are coming as if they are light rays coming in all directions, isn't it? When they pass through condenser lens will modify them into a beam of light. Is this part clear to everyone, beta?
Yes, ma'am.
Yes, ma'am.
This is what is the role of condenser lens. They condense it. The scattered light is focused to form a beam of light. That is what is the role of condenser lens. Now we will talk about the next part of microscope, that is this part. This part is called
Stage.
no.
Stage.
Stage is age. Stage is age. So this is the stage. What is the role of stage?
Ma'am, it holds the specimen.
Very good. It holds the specimen. This part is known as clip. It fixes specimen at a place. वो specimen को एक जगह रखता है. Let me tell you that, beta, this is the type of microscope observed in case of school laboratories. There are other modifications also. The problem with this type of microscope is you require proper sunlight, then only they can work. Okay, because in like night time, if you are having tube light, the image formation is not that good. But the person who is going for research may have to work in lab during night time also. To solve this problem, what nowadays is done, they replace this mirror and in place of it, they put one light bulb. Okay, with the glow of which can be controlled. Okay, so that way microscope can be used during night time also in absence of sunlight. Second modification that is observed is this stage. This stage is a fixed one and the clip is a fixed one. Fixed one, I mean to say, obviously you can open and place your specimen over there. But if you want to move the slide here and there, because what happens is, beta, consider in a slide, at the middle part you have the cells that you want to observe. When you look under microscope, you will be able to see this part only. It will not give the view of this whole part. It will never ever give you the view of the whole part. So if you want to study something and if that part is missing, you have to move that slide this side or this side or this side or this side and search for the whole region for the accurate stage that you want to see. Understood? Because when you are preparing slide, you don't know what is coming to your slide because things are microscopic. Are you getting me what I am saying, beta?
Yes, ma'am.
So in this type of stage, which is simple stage, you have to move it with your hand. And then like we don't have that much precision, your eyes are under the lens and you are moving, it's difficult. So nowadays mechanical stage is available where knobs are there. By looking into microscope, you can rotate the knobs and you can change the position of the slide. Okay. So those mechanical stages are better. In stage, you can see at the central part there is a hole. There is a hole. This hole is there so that light can come in. Okay. Now what are these G? They are
Objective lens.
so what are the types of objective lens?
Ma'am, 10x, 100x, and 45x.
So 10x is called low power. 45x or 40x it is, that is called high power, and 100x is there in some rare cases, that is called oil immersion lens. So what is this X representing?
वो ना मैग्नीफाई करके देख सकता है.
Okay. So before we proceed for the concept of magnification, I wish to ask you two questions. Consider you have an image which is like this of hand and an image which is like this. Two images are there, A and B. Which one do you think is better?
B.
But it's not magnified, no?
Yes.
So always remember in a microscope, it's not only about magnification. If you see a cell which is blurred, you cannot see the outline, you cannot see the internal parts of the cell. Will that image do? बहुत बड़ा image बना के दिया है वो आपको उसने, but it is not good. चलो मैं ये आपके लैंग्वेज से समझाती हूँ. देखना जब हम SLR कैमरास यूज करते हैं, there is a mode called micro mode. उसमें आप किसी चीज का बहुत सामने से अगर पिक्चर ले रहे हो, फिर भी वो चीज बहुत क्लियर आती है. हमारी मोबाइल कैमरा में जितना आप अगर थोड़ी लो क्वालिटी मोबाइल कैमरा लोगे, आप अगर बहुत पास से पिक्चर लोगे तो देखना उसकी क्वालिटी उतनी अच्छी नहीं है. So this terminology is called resolution. So resolution of a microscope should be good. Otherwise, it's of no use. Better. The resolution of microscope is the one of the most important property apart from the magnification. Magnification is required, but apart from that, resolution is also required. So these objective lenses, they are magnifying things 10 times, 45 times and 100 times. So what is this oil immersion lens? Acha, can you tell me what do we mean by refraction?
Ma'am, bending of light.
When it occurs?
When the light is changing, when it's traveling from one medium to another medium.
So consider this is your objective lens. Okay, and this is your specimen. So normally what happens, the beam of light that is coming from condenser will pass through the specimen, and after passing through specimen, before it can go to objective lens, it will have medium as air. So what will happen? Here will be refraction, and only a portion of the light beam can go into the objective lens. Do you agree with me?
Yes, ma'am.
So to solve this problem, there is a way. What is the way? The way is we can layer this area with oil in between. So in place of air, we will now have oil. Generally, cedar wood oil is used. Why? Because it is having similar refractive index of the specimen and glass. Okay. So इससे क्या होगा? The thing that will happen is the light rays which were scattering will not scatter, and objective lens can retain maximum number of light rays that is coming from the specimen. Okay. So this is the beauty of oil immersion lens, and this is the reason for which when high power objective lens is giving you 45x magnification, objective lens that is oil immersion can give you 100x magnification. Is it clear why? What is the beauty of oil immersion lens?
Yes.
Yes, beta.
मैम आपने ये ऑयल इमर्जन का पता है तो सच में ऑयल रखते हैं 100x में ऐसी इमेज दिखती है जैसे ऑयल इमर्जन में दिखती है?
नहीं, इमेज में तुमको ऑयल दिखेगा ही नहीं, इमेज तुमको मैग्निफाइड दिखेगा। उसकी रीजन क्या है? क्योंकि हम लेंस और स्पेसिमेन के बीच में ऑयल की एक लेयर डाल देते हैं। वेरी सिंपल इट इज। आपको बस लाइट लेनी है, बिफोर यू पुट इट अंडर द लेंस you have to put a drop of oil, फिर उस लाइट को लगा के, and the beauty of this oil immersion lens is generally ये lens it can touch the surface of slide. इसकी ये वाली जो part है ना, there is a spring through which the lens can move up and down. Okay, is it clear?
Yes, ma'am.
So you
Yes,
will
ma'am.
not see oil in the under microscope, and you should not see. Your purpose is not to see the oil. Your purpose is to see the specimen more clearly. That is to have that clear image. To stop the refraction, we are using oil immersion lens. Okay, is this part clear to everyone? Chalo. Now we will talk about this one. What is this? This is called arm.
Arm.
Whenever like you are taking microscope from one place to another place, you should always hold the arm and another hand should be at the base. उसको हिलाते हुए ऐसे लेके जाना is not correct. Okay. Now this part, this is the cylindrical part. Many a time prisms are available here. In that case, this upper part will be twisted, and you can look into microscope even after sitting. ये वाले microscope में either you have to twist it towards you, then light का problem होता है, or you have to stand and see. Okay. Next is this lens. This lens is called ocular lens.
Hi. मैम आईपीस लेंस भी बोल सकते हैं ना?
Yes. ये generally 5x और 10x होता है. Very rarely it can be 15x also. So if I am looking something under low power, तो उसकी मैग्निफिकेशन कितना होगा? लो पावर का 10 होता है 10x मल्टीप्लाइड बाय 10x, so it will be 100x, मतलब 100 टाइम मल्टीप्लिकेशन मैग्निफिकेशन. Then for high power it will be 45x multiplied by 10x, so it will be 450 times magnified. And if I am looking for oil immersion, it will be 100x multiplied by 10x, so it will be 1000 times magnified. Is it clear?
Yes, ma'am.
Yes, ma'am.
So apart from this, another thing I wish to say here, always whenever you are focusing any specimen, you should always start with the low power. Once you fix your slide in low power, then you can go to high power and oil immersion. पहले high power या oil immersion use नहीं किया था. These are two knobs. This is known as force adjustment. And this is known as fine adjustment.
Fine adjustment.
So when you are moving them, this whole thing will go up and down, and it will be there for the adjustment of focal length. Okay, because we know the best image is formed in focus. That's why. Okay, so these are parts of microscope. If you wish, you can take a screenshot. Obviously, I'm going to share the video. Any doubts, anyone?
No, ma'am.
So then we will have a look into a very beautiful video. Give me 1 minute to share the screen. Look into the demo of a microscope.
Besto, built on trust. View larger than life. A practical introduction with projects and activities. Microscopes, parts and their uses. A. The eyepiece, ocular, is the part of the microscope closest to your eyes, through which you look. It contains the ocular lens, which makes the image produced by the objective lenses larger. An eyepiece may be labeled with its magnification or power, for example, 5x or 10x, meaning it enlarges 5 times or 10 times. D. The body tube holds the eyepiece in position over the revolving nosepiece and the objective. The body tube may be straight up and down or slanted. C. The arm is the curved metal piece that holds the body tube in place over the stage and the base. D. The revolving nosepiece holds the objectives and allows you to change objectives while looking at a slide or sample. E. The objectives are the parts at the bottom of the body tube closest to the sample you are examining. Each objective has a lens and a tube-like holder, the mount. The longer mounts hold the stronger lenses, the high-power lenses. The shorter mounts hold the weaker lenses, the low-power lenses. Like the eyepieces, objectives also come in various parts. Microscope has several objectives, for example, 4x or 5x, 40x or 45x, and 100x. Some microscopes only have 1 or 2 objectives. F. The stage is the flat surface on which you put your slides or samples. G. The stage clip holds the slide in place on the stage. H. Beneath the stage in all the Besto microscope have a condenser having standard diameter of 39.5 mm which collects and concentrates the light before it passes through the sample. I. The iris diaphragm is used to control the diameter of the cone angle of the light transmitted onto the specimen. J. The coarse adjustment knob is the large knob used to adjust the position of the body tube, allowing you to quickly bring your sample slide into view. K. The fine adjustment knob is the small knob used to change the position of the body tube by making small adjustments to the focus of your sample. Almost all of our Besto microscopes have both coarse and fine adjustment knobs, but some have only one knob. L. The mirror, located beneath the stage diaphragm, increases the amount of light shining through your sample or slide. Sometimes we use lamp plugged into an electrical outlet instead of mirror or reflector. M. The base of the microscope is the heavy bottom part. It supports all the other parts of the microscope. N. Substage adjustment knob is used to move condenser up and down, bringing it into focus. You're probably anxious to get started using your microscope. Before you use your microscope, you need to know how to set it up properly. This video contains instructions for you to follow. You should come back to here each time to use your microscope until you know the instructions by heart as well as the microscope. Step 1. Remove the Besto microscope from its box or cover. Always pick up your microscope correctly. Grasp it firmly with two hands. One hand under the base and another on the arm. Place the microscope on a table, away from the edge. Move it to a position in which you can look comfortably through the eyepiece. You may want to sit on a chair or stool. Step 2. Make sure low power objective is in place over the hole in the stage. Use the coarse adjustment knob to raise the objective so that its lowest bend is about 1 inch, 2.5 centimeter above the stage. In all Besto microscopes, aperture control is by iris diaphragm. At this stage, open it fully. Step 3. Put the slide on the stage so that the center, the bit you want to look at, is over the hole and under the lens. Use the stage clips to hold the slide in place. Step 4. Look through the microscope. You should see a circle of light against a black background. Twist the plano concave mirror around to make the circle as bright and even as possible. If you cannot see a circle of light, your objective lens is probably not in line with the ocular tube. Adjust the nosepiece until you hear the lens click into place. Step 5. The reflector has one side flat, while the other is concave. It is difficult to say which one is best to use in every case. You will have to experiment to see which gives you a better view. The flat side will provide a sharper image in most cases, while the curved side will provide a brighter image. Here's a helpful hint for you. Never use your microscope in direct sunlight, as the reflected light could damage your eyes. Step 6. If you are using a lamp of your lighting, put it about 20 centimeters away from the microscope. Adjust the lamp so that the light points towards the mirror under the stage. Step 7. We have already told you in step 2 that low power objectives is in place over the hole in the stage. Look through the eyepiece and use the coarse adjustment knob to focus upwards or move away from the sample or slide. This should bring the slide or sample into view. If you go too far, simply begin again at step 2. Step 8. When the specimen slide comes into focus, it will become sharp and clear. Step 9. To use the high power objectives, turn the revolving nosepiece to bring it into positions over your sample or slide. You should be able to see the sample or slide through the high power objective, so you should only have to adjust the fine adjustment knob. Step 10. Now adjust the aperture control with the help of diaphragm. Close it until circle of light dims and then open it again slightly and stop it where you get maximum resolution of slide or sample in view. Step 11. Medical microscope model Besto 10 have a special type of lens called an oil immersion lens 100x. This lens is used by placing a drop of oil, cedarwood oil, on the top of the cover slip and adjusting the position of the front lens of the objective so that it is touching the oil drop. The oil bends the light more than the air wound, so more light can enter the microscope. All immersion lenses make it easier to see samples because they increase the amount of light going through the sample or slide and microscope and into your eye. Convert your simple microscope into LED illumination microscope with LED light source. Our Besto LED light source eliminators, the variation that due to reflector light or hologram lamp technologies. Features, some interest.
So how was the video, class?
Nice, ma'am.
Nice one.
You can actually keep it as a reference. Maybe someday when you will be able to go to lab, you can use them because this is something which is very, very important for you and you have to learn about it. Okay, now quickly I wish to talk about some very important thing, that is about the different images like I wish to share. But do you think light microscope is the only microscope that is available? Class? Any idea about different types of microscope we have?
Ma'am, electronic
It
microscope.
is compound. It's called compound microscope. Apart from compound microscope, there is simple microscope. Compound is called compound because we have only one set of lens present of there. Okay, that's why it is called compound microscope. And in case of like light microscope, simple and compound, these are the two variations we have. But let me tell you, beta, other than these various source of lights like UV light, there can be phase contrast microscope, there can be electron microscope, because electron microscope is something which revolutionized the concept of microscopy. It is having a very high ability to look into tiny things from the atomic level. Okay, so we will not go in much detail of that. I wish to share with you today now the thing that is spotting thing. I wish to share with you a few images. You try and identify it. Okay. So this is the one. Can you identify what it is?
Mushroom or basically fungus.
Mushroom.
I want
Yes,
the
ma'am.
name.
Basidiomycetes.
I want the name.
मैम अगारिशियस ऐसा कुछ है ना?
Agaricus.
हाँ वही.
Agaricus bisporus. Okay, and it is Deuteromycetes. That is correct. So this is the way I was expecting you to answer. Can you identify this one? What's image is this?
Bacillus.
Okay, so it is Bacillus, you think. Bacillus बोलने से चल जाएगा? What it is?
Bacteria.
It is a bacteria. Okay, so that is correct. It is a bacteria. Now we will look into the next one. What is this?
मैम स्पाइरेलियम है
Check.
मैम ब्लर दिख रहा है
मुझे आपको वो दिखाना था. I could have given you image which are drawn. Actually, let me give me one moment to switch on the recording. I think we are recording the thing. Yes, we are. When you look them under microscope, they look quite different. And I wanted you people to look into that picture only. Tell me what it
मैम
is.
ये क्या है
That I am asking to you what it is. It is
मैम
coccus.
मुझे लगा अभी आपने आंसर बोला
It is coccus. It is coccus. Spherical bacteria. Okay. This is the spherical bacterium. I wish to share with you a few more image of bacillus.
मैम ये तो कॉकस है ना जो अभी आपने शो किया
वो तो कॉकस ही है.
मैम बट वो कॉकस जैसा तो बिल्कुल नहीं लगता.
हाँ, they are
मैम वो स्पाइरेलियम जैसा दिख रहा था कुछ
हाँ मैम.
expecting them to be having perfect shape. They are not big. You are looking. What is this?
Bacillus bacteria.
This is a clear image of the
bacteria.
Okay. So now we will move ahead. Tell me what is this?
मैम ये जिम्नोस्पर्म है
मैम राइजोपस
राइजोपस
Is
मैम
not-
राइजोपस है
Yeah, gymnosperm. What is this?
राइस
Right.
Yes. So it will be rice है मतलब rice बोलने
मैम राइस
it is an angiosperm monocot.
हाँ yes मैम.
Yes.
What is this?
मैम रूट
Rose. Rose.
It is pea plant.
Ma'am.
It is
मैम रूट है
and it is a dicot. What is this?
Spiralium.
Very good.
स्पाइरेलियम
What is this?
Spirogyra.
स्पाइरोगायरा
Chalo. This is herbarium of this. What is this?
Yeast.
यीस्ट
Yes, it is having budding also. If you see, they have budding. So yeast budding structure is also there. Now let me show you. What is this one?
फॉन
Okay. It is fern, isn't it? Which fern?
Peridophyta.
Arey baba, haan, Dryopteris. This is
moth
मॉस
Funaria. You need to
ब्रायोफाइटा
Bryophyta.
Bryophyta.
Their name is Funaria. What is this?
ये क्या है मैम
वही तो पूछ रही हूँ. These are all part of your syllabus. That's why I thought let me at least have a few people a look. क्योंकि when we see the diagram ना, draw किया हुआ diagram and the image actually how they look under microscope, it's quite different. This is oscillatoria, beta. It is another algae.
मैम ये क्या है ब्रायोफाइटा टेरिडोफाइटा ये क्या है
ना, this is oscillatoria algae. Thallophyta है ये.
ओके मैम थैलोफाइटा
Okay, so we are done with the spotting of plant kingdom. Now we will have So after spotting of plant kingdom, we will have a spotting of animal kingdom too. And let me share the screen for that. This will be comparatively easy for you to understand. What is this?
Amoeba.
So this is a image created by phase contrast microscope. What is this?
Ascaris.
So what is like ascaris is?
Ascahelminthes.
Or nematoda. What is this?
Earthworm.
Falling under?
Annelida. Frog.
Frog falling under?
Amphibia.
Another frog.
मेरे वापस frog ही है.
Honeybee.
So it is under
Arthropoda.
arthropoda. Very good.
Arthropod.
These are three types of honeybee you can see: worker, queen, and drone. What is this?
Hydra.
It is falling under?
Nidaria.
हाँ मैम Nidaria.
Superfluke.
Very good. It is falling under?
Platyhelminthes.
Platyhelminthes.
Pigeon ave.
Ave.
Great.
Prawn.
Prawn.
It is under?
Arthropoda.
Very good.
Rabbit mammal. Yeah.
Fish.
Hmm.
Fish, so it is coming under which? What type of fish is this? Class? Is
Ma'am, एक minute.
Ostenthius or Condrectis?
Ma'am, Ostenthius.
Yes, Ostenthius. How you identified Rashmi?
मैम वो कल आपने नोट में लिखवाया था ना.
क्या लिखा था? वही तो सुनना है.
मैम वो उसका माउथ की जो पोजीशन है वो वेंट्रल साइड पे होती है.
And tail,
इसका
tails are also, they have big
tail
scales.
dorsal में होती है और face ventral में होता है.
Great, great. So, what is this?
Shark.
Falling under?
Chondriocytes.
Very good. This one?
Silkworm.
So, this is actually the
Annelida.
larva stage of the silkworm. I wish to show you, let me first like finish with this, then I will show you how the insect looks. This one?
Snail.
Snail.
Polyester?
Mollusca.
Shabash. This one?
Starfish.
Falling under?
Echinodermata.
So this is the ventral view of starfish. This one?
Leech.
Annelida.
Great.
Lizard.
Reptile.
Wall lizard. You have to say wall lizard. Lizards are of very years time. Beta. And this is under what? Reptiles. Isn't it?
Yes, ma'am.
Yes.
So all of you can see one more specimen. Look into the mirror. Can you tell me it is falling under which super class or class? When you
Mammalia.
all of us. So for that we don't require any specimen. So this is a fun way by which we revise the chapters. But still, you need to remember the characteristics of each and every organisms. Okay, you cannot take a risk of forgetting them. I promised you, so I will show you the silk moth, how they look. I will give you a glimpse of that. Any other doubts after having a what I can say flashback? Any doubts that came in into your mind? Class, chalo, I will show you the image of. See, this is how they look. But we don't actually allow them to grow. We actually extract silk. For extraction of silk, the larva needs to be killed. So we are greedy, obviously. So this is how silk moth look. Bombix mori. Is my screen visible?
Yes, ma'am.
So that's all from my side about animal and plant kingdom. So next day, when you propose, when you are ready, we can have quickly a quiz, whichsoever day you decide, and we can make sure you have systems and good internet connection so that you can play a quiz. Anything else from anyone? Class?
No.
मैम
Yes, beta?
मैम जैसे आर्थ्रोपोडा लार्जेस्ट फाइलम है तो ऐसे स्मॉलेस्ट या वो फाइलम कौन सा है बता सकते हो मैम आप?
It is actually never asked. I think it will be porifera. Let me check and get back to you.
ओके मैम.
Any other question? Very valid and good question. Anyone, any other question? So we will say a bye here. We will meet in our next class and discuss further. So I am going to start chapter number 5, that is morphology in the plant. So in our next lecture tomorrow, we are starting with the chapter. Okay, class?
Yes, ma'am.
Okay.
Yes.
So then bye to all of you.
Bye.